Hello there. This is the walkthrough video for Friday. It is the 26th in Try 1, Week 8. Hello. Using one or more good sentence, can you please answer the following? Why are cells sized and shaped the way they are? So you can go back and look at your cell size lab, which might still be in progress. We'll be debriefing that on Monday. You might look at your notes for 2.2. You might look in your review book about investigation 4, because this water potential suite of labs we've been doing is actually part of the official AP Bio lab manual from some years back, and it's in the back of your review book under investigation 4. Maybe you probably definitely need to talk about surface area to volume ratios. And if you don't know what's happening, you can go ahead and take a look there. So today's the same, SAME. We're observing osmosis in living cells, and we're going to apply what we've learned from our 2-cell models and our work together. So today in class, your colleagues worked on the water potential lab for day 2. So if you weren't here yesterday on Thursday, I gave folks some potatoes and I gave them some solutions, and I said, which solution is which? I've mixed them up. I didn't mix 2 of them up, but I mixed the rest of them up. How will you know which one is which? And the answer is people cut some potato, they measured the initial mass, they incubated them in the different solutions overnight, they measured the final mass, and we're going to calculate percent mass change. Now, if it turns out that you do not have the correct assignments, you will have trouble making quantitative measurements of water movement. So if you have not seen data from your colleagues, you will need to touch base with them in order to complete this lab. But you're going to write in your lab notebook, first question, which sucrose solution is which? And then you're going to write a sentence or 2 to answer, including how you know. And I want to remind you that plant cells might gain mass or lose mass, depending on the types of solutions they're placed in. Now, unfortunately, we cannot use these phrases because do you remember that plant cells push against their cell walls? So we can't use hypotonic, isotonic, and hypertonic, but the same idea applies, right? So the next question, you're going to copy this in your lab notebook, and we actually made a graph today. I'm going to show you what it looks like. So you've been making a lot of graphs that look like an L. We're not doing that today. Instead, you're going to make this T graph, and where the T is going to depend on the mass changes that your lab mates will give you the data for. We made an axis that looks like this. Then we graphed our points. I will tell you that the blue solution is 0.2 molar and the yellow solution is 1.0 molar. I'm not sure which ones these are. Your data might vary. And I plotted those data points. Now, the next thing I did is I got out a ruler, and instead of doing a complicated calculation like you would do if we were really doing it, we just tried to eyeball a line where the amount of space above and below the dots was approximately equal. Good enough. And you're going to find an x-intercept. It's going to be different from the one that your colleague makes because your line will be slightly different. So now you're going to label on your graph where that x-intercept is. And indeed, it's at 0% mass change. If you draw a best fit line through the percent mass changes on a graph of sucrose concentration versus percent mass change, the x-intercept is the imaginary sucrose solution that would have yielded a 0% mass change. Part 3, you're going to copy this question. This is what we're answering in class on Monday. If you are still feeling in the middle of the things, that's okay. So are a lot of people who are here today in school. Hang in there, come to class on Monday, and I will see you then. Today, we skipped the cool down. We went on to the weekend review. Here's some content to help you think about that. Hope everything is well. Take care out there. I'll see you Monday. Bye.